Good afternoon, everyone. Now, just to begin, I'd like to acknowledge the traditional owners of the land, the Wurundjeri people of the Kulin Nation, and to acknowledge and to pay my respects to their elders past, present and emerging. Now, it gives me great pleasure to introduce Andrew to you this afternoon as he tells you about the work that he's been doing over the past 3.5 years as part of his PhD. I first met Andrew 5 years ago now. At that time, he was in the final year of his science degree at Melbourne University and was thinking about what sort of honours project he might like to do. At that time, it was already pretty clear that Andrew liked to follow his own path and do things a little bit differently. So, for example, rather than going straight from high school through to university, Andrew spent a number of years originally working for his grandfather's local newsagent business as he tried to figure out what it was that he wanted to do in life. As it turns out, Andrew wanted to become a scientist, and it's been my great privilege to be involved in that journey over the last few years. And once again, I think through that journey, Andrew has shown his ability to do things a little bit differently, as I'm sure you'll appreciate over the next 45 minutes or so as he tells you about his work. So with that, Andrew, over to you.
Thanks, Ken. I'm not sure if being different is always good. Hopefully in this case it has been. This is one of my favourite figures. It's from an article published about 40 years ago. It was written by a senior rheumatologist, and it was a list of cures for rheumatoid arthritis that he encountered throughout his career. And some of them make sense. For example, chemotherapy. Methotrexate initially began its life as a chemotherapeutic agent, and it's now used as a frontline drug in the treatment of rheumatoid arthritis. Some are obvious. Everyone knows gin's a panacea. But my favourite is standing inside of whales, specifically dead whales. I'm curious to know what the mechanism is for this. Could be a potential honours project. So this is the outline of my talk. I'm going to talk, first I'll introduce rheumatoid arthritis and extracellular vesicles, and I'll move on to work I've been doing isolating extracellular vesicles from human synovial fluid, followed by characterising these synovial fluid extracellular vesicles. And finally, if time permits, I'll talk about work we've been doing in trying to predict response to therapy in patients with rheumatoid arthritis. Rheumatoid arthritis is a systemic chronic autoimmune disease characterised by inflammation in the joints. It affects a large portion of the population, and it can result in severe disability. While genetic and environmental factors are implicated, we still don't completely know the underlying causes of the disease. Rheumatoid arthritis is thought to arise in genetically susceptible individuals following environmental triggers, and these are thought to include smoking and periodontal infection. Following this, this leads to a loss of tolerance of modified self-proteins, causing an inflammatory response, which for reasons unknown is localized in joints. The inflammatory response in joints is perpetuated by a number of positive feedback loops which lead to ongoing synovitis. And the conditions are associated with a number of coexisting conditions which likely exacerbate the disease, and some are indicated there. A number of drugs are now used to treat rheumatoid arthritis. These include steroids. The conventional disease-modifying anti-rheumatic drugs are a frontline treatment, and these are some of the key ones are listed here. These are methotrexate, sulfasalazine, and hydroxychloroquine. We don't really know how these conventional DMARDs work, but they've been employed for a few decades now, and they're generally, quite effective, and they're cheap. So this has led to them persisting as frontline drugs in the treatment of RA. More recently, targeted biological agents have been developed, and these are generally monoclonal antibodies which target specific proteins involved in driving rheumatoid arthritis, such as CD20, which is involved in B-cell activation, and key drivers of inflammation such as TNF and IL-6. And more recently, small molecule synthetic inhibitors have been developed, which are small synthetic compounds which target specific proteins such as JAK kinase inhibitors. But it's important to note that all these drugs really while they're effective at controlling the progression of rheumatoid arthritis or controlling inflammatory symptoms, they don't cure the disease. And it was stated, I think, really quite nicely by Ian McKinney and George Shett that molecular emission and the capacity to re-establish immunological tolerance remain elusive. So there is no cure for rheumatoid arthritis that we're currently aware of. Extracellular vesicles, or EVs, are lipid bilayer-bound vesicles. They're released from all cell types and found in all bodily fluids. By EVs, I'm referring to exosomes and microvesicles. The exosomes originate in late endosomes, and following fusion of the endosomal membrane with the plasma membrane, the exosomes are released into the extracellular space, whereas microvesicles arise from the direct outward budding of the plasma membrane. Now, both exosomes and microvesicles contain a cargo of RNA and protein, which, upon interacting with recipient cells, they can elicit function or functional effects. And they're thought to do this via a number of different mechanisms. This includes through protein-receptor interaction, whereby an EV membrane protein can interact with its cognate receptor on a recipient cell, leading to signaling effects in the recipient cell. EVs are also proposed to fuse with recipient cell membranes, causing release of their contents into the recipient cell. Similarly, EVs can be endocytosed, and the EV membrane and the endosome membrane is thought to fuse, releasing the EV contents into the recipient cell cytosol, where they can elicit functional effects. Endocytosed EVs also can also undergo degradation of the EV membrane, causing release of the EV contents where they're free to bind endosomal receptors and initiate signaling effects in the recipient cell type. And this is a nice short video put together by the NIH showing a malignant cell releasing large numbers of EVs, and they're being taken up by a recipient cell and inducing the transformation of that recipient cell. And there's a number of now proposed roles for EVs in rheumatoid arthritis. And to illustrate, here is a schematic of a synovial joint, a rheumatoid joint. I have the bone here. The bone is protected by cartilage, articular cartilage, which consists of chondrocytes. Here is the articular cavity, and this contains synovial fluid. Some token neutrophils are present there. Generally, they're found in high numbers in synovial fluid in rheumatoid arthritis. And here is an inflamed synovial membrane. We have the inner layer, which consists of fibroblast-like and macrophage-like synoviocytes, as well as the subintimal layer, which contains a range of different infiltrating immune cells. Now, all these cells release EVs, and they're found in high numbers in rheumatoid arthritis synovial fluid. And it's been shown that EVs in RA synovial fluid can stimulate recipient fibroblast synoviocytes to induce release of pro-inflammatory cytokines and chemokines. And this is proposed to lead to angiogenesis, further immune cell infiltration, and we propose that this effectively creates a positive feedback loop contributing to the perpetuation of joint inflammation. On the other hand, EVs released from TNF-stimulated neutrophils are described to migrate into cartilage and promote anabolic and anti-inflammatory effects in recipient chondrocytes. Despite all this, there's still much we don't know about what EVs are doing in rheumatoid arthritis. We don't have a clear understanding of what cells are the major EV generators. We don't have a comprehensive understanding of what's in the synovial fluid EVs. We don't know, we don't have a great understanding, again, of what changes in disease or how EVs contribute to RA pathophysiology. So these are questions I wanted to investigate in my PhD. And here's a beautiful image from a lab in the US showing an activated cell releasing large numbers of EVs. Okay, so first, to investigate EVs, I had to obtain an isolation of EVs to work with. I had to purify them from synovial fluid. When a rheumatoid arthritis patient presents at the hospital, the clinic, rheumatologists will sometimes have to aspirate their synovial fluid as part of routine clinical care. And if the patient consents, then I will get a call and I will go and collect the synovial fluid. Once I get the synovial fluid, the first thing I do is I deplete the cells, and then I aliquot it out. My next step following aliquoting out the cell-depleted synovial fluid is to separate out the EVs from the other material in the synovial fluid. Now, synovial fluid is a very viscous, complex fluid, and apart from EVs, there's many other components in it. There's high levels of hyaluronic acid and proteoglycans, and these typically function to reduce stress upon movement. There's also carbohydrates, fatty acids, and DNA. So you can imagine separating out the EVs from all this other material is a bit of a challenge. Traditional methods of EV isolation employ differential ultracentrifugation. So more or less what happens here is we obtain the cell-depleted, so following depletion of cells, the supernatant is then spun again at 10,000 Gs to deplete remaining extracellular debris in apoptotic bodies. Then the supernatant is collected, spin that at a high speed to obtain a crude EV palette. This is washed and then spun again at 100,000 Gs to obtain the EV-enriched palette. A more stringent purification employs a sucrose density gradient. So here, the crude EV palette is overlaid on a discontinuous sucrose gradient, and then this is spun at a high speed overnight. And the theory behind this is that EVs should separate out from contaminating material based on differences in their buoyancy. However, published in the literature, people who employed these methods have described unsatisfactory results. And here's an example employing differential ultracentrifugation. While this method is capable of purifying EVs, including this really happy guy, there's also numerous contaminating or amorphous material, as indicated with the asterisks. Studies employing a sucrose gradient also suffer from poor quality isolations in that there are definitely EVs there, but there's considerable amounts of contaminating material, as indicated by the white arrows. So in more recent times, people have started to use size exclusion chromatography as a method for EV isolation, and good quality enrichments have been described in various biological fluids, including blood plasma and urine. And this method is thought to have advantages to ultracentrifugation-based methods in that higher yields of EVs are described, and it avoids aggregation and vesicle rupture, which is associated with high ultracentrifugal forces. So we look to investigate this method. Essentially, just size exclusion chromatography involves a column which is packed with a porous bead matrix, and these are some beads illustrated here, and these contain pores. The pores are quite small, and only small material can traverse into the pores, whereas large material can't access the pores. So larger material has a shorter path to go through and it elutes quicker, whereas smaller material, such as the proteins, are small enough to access the pores, so it takes longer to elute. So what I do is I inject my synovial fluid into the column. It then flows through. The EVs elute first, and I capture them, and then I discard the remaining flow through. I then look to compare different methods and assess their quality of EV isolation from synovial fluid. And you don't need to worry too much about the details here unless you're really interested. Essentially, what I did is I compared differential ultracentrifugation, sucrose gradient ultracentrifugation, and size exclusion chromatography. First, I will go through the ultracentrifugation characterization. So here we saw the presence of EV markers as expected, but when we looked for the presence of common contaminants, such as serum albumin, which is an abundant plasma protein, apolipoprotein A1, which is present in high-density lipoproteins, and fibronectin, a constituent of the extracellular matrix, we were unable to deplete them from EV isolations using standard ultracentrifugation-based methods. I further characterized this isolation with electron microscopy, and we saw images similar to what had been published. We were easily able to see EVs, but there were considerable amounts of contaminating material as indicated by the white arrows. So this method was unsatisfactory for our purposes, which was to accurately identify the contents of EVs. Similarly, with a sucrose density gradient, we saw EVs eluting having buoyancy at a density between 1.12, pardon me, and 1.19 grams per mil. However, we were unable to separate the EVs from much of the high-density lipoproteins or the fibronectin. We did see some separation from the serum albumin, though. And then I took fractions, so these fractions with densities ranging from 1.12 to 1.19 and further assessed them by electron microscopy. And again, we saw the presence of EVs, but there were still high levels of contaminating material. So this method was also unsatisfactory. So employing size exclusion chromatography, so following size exclusion chromatography, you get an A280 profile of the elution. The A280 profile is a measurement of the protein which is coming out of the column. We see that the majority of the protein is eluting from fraction 5 onwards. So I should mention I collected 8 different fractions at these different time points. And we saw, again, the protein coming out from fraction 5 onwards, but I then collected each fraction and concentrated them by ultracentrifugation at 100,000 Gs and resuspended them in an equal volume of lysis buffer. Then to investigate which fractions have protein which is being pelleted at 100,000 Gs, I ran the different fractions on a gel and found that protein was being pelleted across fractions 2 to 7 via 100,000 G ultracentrifugation. I then investigated where are the majority of the EVs eluting, what fractions are the majority of the EVs eluting, and I did this via volume normalized loading, and this indicated that the majority of the EVs are coming out between fractions 2 to 4, as judged by the prevalence of EV markers. But what about the contaminants? So serum albumin, most of the serum albumin is coming out from fractions 5 and 6. We saw that with apolipoprotein A1, it's coming out from fractions 4, 5, 6, and 7, and most of the fibronectin is coming out in fractions 4 and 5, or 5 and 6. And then I assessed the purity of the isolation using protein normalized loading, and we saw that in fractions 2 and 3, we're able to obtain EVs, but with minimal amounts of contaminating serum albumin and apolipoprotein A1. There was still the presence of some fibronectin there, indicating that there is some remaining contaminating fibronectin, or the EVs might, to some extent, be associating with the fibronectin. So these looked good, much better than the other methods. So I further characterized these by electron microscopy. We saw there was considerably less contaminating material than based on the other, than when compared to the other methods, but there was still quite a bit of contaminating material remaining. Some of it does appear to associate with EVs, some of it doesn't. So again, though, this was still not ideal. So we thought, what if we take this isolation and we treat it with proteinase K, which should theoretically digest all this extracellular contaminating protein, but the protein inside EVs should be protected by the lipid bilayer membrane. That's what we did. And I compared proteinase K treated isolation with a control isolation. So this was size exclusion chromatography eluent from fractions 2 and 3. We found a vast reduction in the amount of protein with proteinase K treatment, and I've compared the untreated and treated here, and with the electron microscopy analysis indicated that there was a robust reduction in the amount of contaminating material, but EVs were still present. So we were quite happy with this. So to summarize, size exclusion chromatography we showed was capable of depleting contaminating material, specifically contaminating HDLs and albumin from synovial preps, but there was still considerable amounts of contaminating material present. And with proteinase K, we could further deplete the amount of contaminating material. Okay, so we had a high quality isolation. We thought this was good enough to go forth and characterize and accurately characterize and comprehensively characterize the protein and the RNA content within the synovial fluid EVs. To do this, I used a cohort of rheumatoid arthritis patients obtained from the Royal Melbourne Hospital. There were 7 patients with high disease activity and 5 with low disease activity. So their disease activity was determined based on the number of synovial fluid white cells. So RA patients who have high levels of white cells in their synovial fluid typically have greater inflammatory processes, and with lower numbers of white cells, the disease is generally less severe. So we took these EV isolations and we subjected them to proteomics and RNA-seq. So first I'll talk about the proteomics, and this is work we did in collaboration with Laura Dagley. The first thing I did was following the isolation of the EVs, I assessed how much protein's in them. Is there more protein in the EVs in patients with high disease or low disease? So I took the EV isolation and normalized it to amount of synovial fluid. I found that there was a trend to there being more protein in patients with high disease activity. I say trend, it was a p-value of 0.07. It didn't reach that magical cutoff point. I think with more numbers, it probably would. Then assess the profile using a gel electrophoresis, and this is a silver stain, investigating the different protein profiles in between high inflammation and low inflammation. We see that patients with high inflammation have more bands, a more complex EV profile or protein profile in their EVs compared to patients with low levels of inflammation. And this was supported by our proteomics results in that we observed about a high number of proteins only in the patients with high levels of inflammation. So I next asked, where do the synovial fluid EVs originate from? What cells are they arising from? To do this, I took the proteomics data, the list of proteins, and I manually went through it to investigate the presence of specific cell lineage markers. What we found is that in both patients with high and low disease activity, neutrophils and fibroblasts appear to be the major EV donors. In patients with high disease activity, however, we see a greater appearance of markers of T cell, dendritic cell, erythrocytes, and other immune cells. Interestingly, we didn't detect any EVs from B cells or platelets, and there was not a robust detection of EVs of monocyte macrophage origin. So this doesn't mean that they're not there, it just means we didn't detect them, and this could be due to there being the peptides here being masked by high levels of other peptides, including markers of neutrophil and fibroblast origin. I then performed differential expression analysis between the high and low disease activity patients. I found that there were 415 proteins which were significantly upregulated in patients with high disease, whereas only 6 were increased in patients with low disease. This is an MA plot, and I'm going to show a few of these, so I'll just walk through it. So each dot refers... Literally. I didn't mean that. Okay. So each dot refers to a protein. The y-axis refers to the log2 fold change. A positive value refers to an increase in patients with high disease. A negative fold change refers to an increase in patients with low disease. And the x-axis is a measure of their abundance. And the ones which had an adjusted p-value less than 0.05 are coloured in red. So I first looked at the 6 proteins which are elevated in disease. And one which immediately jumps out is the HLA-DRB4 protein. This has known involvement in rheumatoid arthritis. It is a component of the MHC2 complex, which is important in antigen presentation. And so looking at this, it didn't really make sense. I thought, why would patients with low disease activity have increased antigen presentation? If anything, you would expect them to have decreased antigen presentation. And going through the data, actually, we saw that there were quite a number of MHC2 components in the data, and most of them were increased in patients with low disease activity. So, this reflects what we would expect, that being patients with high disease activity have increased antigen presentation in their EVs. The other one which jumped out is versicin. So just a bit of background. here is a cartoon showing the composition and structure of articular cartilage. We have bone and then the different layers of the cartilage. So the proteoglycans form an essential component of cartilage, and they make up the structure of cartilage. And one of the major proteoglycans is vesicles. So it's interesting that this is in EVs, and it's tempting to speculate that EVs might have a role in trafficking and disposition of cartilage components. So there were 415 proteins which were elevated in patients with high disease activity. I'm not going to go through all of them. I just thought I'd pull out some of the ones which were highly abundant and had a significant fold change. So taking an arbitrary cutoff of a fold change of 2 to the 5 and an abundance of 1 million. I've selected these 6 proteins. And these, and they're listed here, full-length protein names, and these have defined roles in inflammatory processes. MPO was really highly upregulated in high disease activity, and this is a potent neutrophil chemoattractant, which has roles in bringing inflammatory cells into the joints. CD45 is a pan-leukocyte marker, and it has roles in T and B cell activation. Beta-2 microglobulin is a component of MHC1. RAP1B and actin have roles in cell adhesion and cell motility. One thing which I was quite interested to see was this G protein subunit beta-2, and this is a core component required for GPCR signaling. And looking at the data, look to see if there are other G protein units there, and there were actually quite a few G protein associated proteins in the data. And furthermore, there were a number of GPCRs in there as well, including chemokine receptor 1, as well as receptors for leukotriene B4, platelet activating factor, and complement C5 alpha. And these have important roles in driving inflammation. And furthermore, these RA synovulid EVs contained other key drivers of inflammation, including TLR2, SYK, KRAS, FAS, even IL-17 receptor A, as well as their cognate components which are required for mediating the signal, JAK1, JAK2, TYK2, and STAT1 and STAT3. So this indicates that these EVs have the machinery required for activating recipient cells. There are also a number of anti-inflammatory proteins in there. Annexin A1 was the protein required for the protective effects in recipient chondrocytes. The anabolic and anti-inflammatory effects in recipient chondrocytes mediated by neutrophil EVs was imparted by annexin A1. CD39 and CD73, these are ectonucleases which are involved in the production of extracellular adenosine, again having important anti-inflammatory effects. And TGF-beta receptor is involved in a number of different anabolic effects as well. So I next took those 415 proteins which were upregulated, and I further investigated what pathways are involved in using a program called Ingenuity. And I found that there was a strong association for pathways associated with cell migration. So this data indicates a broad range of roles for EVs, but mostly with a surplus of molecules associated with inflammatory and chemoattractant processes. So next, I characterised a size exclusion chromatography eluent, and this was done in collaboration with Stephen Wilcox and Alexandra Garnham. Again, I looked to see, is there more microRNAs, in the synovulid EVs, and we observed that there was more microRNA per mil of more EV microRNA per mil of synovial fluid, and it was more abundant in patients with high disease activity. A differential expression analysis revealed that there was 49 microRNAs increased in patients with high disease activity and 29 increased in patients with low disease activity. Here's a heat map showing expression of the individual microRNAs in the different patients. And again, an MA plot, much like before, but each dot this time referring to a microRNA. So to investigate what these microRNAs are doing, I again used Ingenuity pathway analysis, and the the experimental outline I took was I took those microRNAs which were significantly up or down regulated, subjected them to, using ingenuity, I identified what their experimentally observed mRNA targets are, so ones which are in the literature, and then I performed pathway analysis on them. So first, focusing on those microRNAs which are increased in patients with low disease activity. The top 5 pathways were shown here, and the one which jumps out is the role of osteoblasts, osteoclasts, and chondrocytes in rheumatoid arthritis. So I thought, what are these? What specific proteins or what specific mRNAs are these microRNAs regulating? And the mRNA targets are listed here, and these are predominantly pro-inflammatory proteins. And it was interesting to see that 2 key drivers of inflammation, TNF and IL-1 beta, were there. So this indicates that in low disease activity, there are microRNAs within the EVs which are controlled in, which are involved in controlling the propagation of inflammation. So next, taking those microRNAs which were increased in patients with high disease activity and performing pathway analysis on their experimentally observed targets, we found a few different involvements, different roles. This time they were the highest, one with the greatest p-value was the role of macrophages, fibroblasts, and endothelial cells in rheumatoid arthritis. I investigated what are the proteins, what are the mRNAs they're targeting. There was quite a few of them, and with the exception of a few, these are all drivers of inflammation. And so this suggests that that microRNAs are also involved in negatively regulating inflammatory symptoms in patients with high disease activity as well. So to summarize, I've shown that neutrophils and synovial fibroblasts are major producers of synovial fluid EVs in rheumatoid arthritis. There was an overrepresentation of pro-inflammatory proteins in EVs in patients with high disease activity. And the protein cargo suggests that these EVs have roles in activating recipient cells and driving inflammation. There are also some key anti-inflammatory molecules suggesting EVs do have some protective role, and this was further supported by the microRNA cargo indicating that these EVs also have key negative regulating roles of joint inflammation. So this is all well and good, but what does this mean therapeutically? What's the point? And I envisage that the data we have, obviously it would need to be validated in a larger cohort, but the data we have might provide rationale for therapies which might be able to mop up or stop the release of these pro-inflammatory EVs. On the other hand, we've characterized some anti-inflammatory molecules which likely have roles in preventing disease. Could we mimic these? Could we create EV mimics or synthetic lipid vesicles which contain this EV cargo? And if these were administered to patients, would they have therapeutic effects? So this is, I guess, where we hope, the research we have will lead. So finally, I'm going to talk about predicting response to triple therapy in patients with rheumatoid arthritis. So the impetus for this project began with work done by 2 of my supervisors, Leslie Cheng and Andy Hill. They published a very nice paper showing that circulating microRNAs could be used to differentiate healthy controls from people with Alzheimer's disease. And what was very nice is that they showed that their microRNA panel showed promise in predicting patients, nominally healthy patients who are likely to go on and develop Alzheimer's disease at a later stage. So we looked to adapt their method to answer an important question in rheumatoid arthritis, and that is, can we predict response to treatment in RA patients prior to them being given treatment? So back to triple. So we looked at a cohort of patients treated with triple DMARD therapy. So triple DMARD therapy involves the simultaneous administration of methotrexate, hydroxychloroquine, and sulfasalazine. These drugs are often very effective, but there's still quite a large portion of patients who don't achieve remission with this therapy. And so those patients who don't respond well can, we can't tell at the outset, we can't pull them out and say, you're not going to respond well, let's put you on another drug. Typically, we just give them triple DMARD therapy because it works a lot of the time and we don't have a better drug. But if we could identify those patients who are unlikely to respond prior to giving them therapy, could we put them on a more effective drug at the outset? So we look to see, are there circulating microRNAs which would help us to do this? And so again, the question we asked was, can circulating small RNA expression predict response to triple DMARD therapy in treatment-naive RA patients? To do this, we collaborated with Susanna Proudman at the Royal Adelaide Hospital, who has set up a rheumatoid arthritis tissue bank, which contains serum samples from rheumatoid arthritis patients collected prior to commencing treatment. And they've all been subsequently treated according to a standardized treatment regime involving triple DMARD therapy, and their response status is subsequently known. Here's a comprehensive list of the patient data. You don't really have to worry about that unless you're interested. Essentially, we classified the patients as responders or non-responders. We had 24 responders and 18 non-responders. The experimental pipeline is illustrated here. Following collection of serum, we extract the RNA. I'll just point out that in this project, we extracted all circulating RNA, not just extracellular vesicle RNA, all circulating RNA. And from a biomarker perspective, we're not too concerned about where the RNA comes from. We just want to identify RNAs capable of obtaining our aim, which is predicting response to treatment. So we had exosomal RNA, we also had free circulating RNA, as well as RNA likely associated with proteins, which is in the circulation. So following RNA extraction, we create cDNA, and then it's subjected to next-gen sequencing and aligned to the genome. And then we look for differences between the two patient groups with bioinformatic analysis. For this project, we used the database of small human non-coding RNAs. So apart from microRNAs, this contains annotations for a number of other small RNAs. And following differential expression analysis, we identified 5 small RNAs which were significantly dysregulated following adjusting for multiple test correction. The 5, so these are listed here. There was 1 upregulated in responders and 4 which were increased in non-responders. So the 1 upregulated in responders is a 5S RNA, and this is typically associated or thought to associate with ribosomes. We also identified in U2 RNA, which is a component of the spliceosome involved in mRNA processing. This was increased in patients with low disease activity. We identified a pi RNA. It's not really known too much about this specific pi RNA transcript. It's poorly characterized. Pi RNAs are typically thought to have roles in retrotransposon silencing. So why these RNAs are decreased or increased in responders, non-responders is of interest, but it's difficult to investigate because we don't really have a clear idea of what their roles are in the first place. 2 ones which caught my interest are these 2 tRNAs. As most of you would know, tRNAs have important roles in bringing in amino acids for protein synthesis. However, looking at the RNA sequence, the tRNAs which we identified weren't full-length tRNAs, they were tRNA halves, and both of them were cleaved prior to the anticodon loop. So these have only really recently been described. They're known to be conserved across eukarya, and they're induced upon cellular stress. And they're thought to have roles in repressing protein translation and inhibiting apoptosis. But why are they increased in patients with high disease activity? I mean, they might just reflect increased levels of cellular stress in patients in non-responders. But I mean, that we don't know that. I suppose what would be interesting is if they might reflect a particular synovial subtype or particular type of arthritis, and this would be interesting to further investigate. Okay, to summarise, we showed a candidate small RNA panel that is indicative of treatment response, However, I mean, for confidence, we would really need to validate this in an independent cohort. And these studies typically require hundreds and thousands or thousands of patients for confidence. And we have not had access to that number of samples to do that. And the small number of samples also provides considerable limitations in that interpatient and technical variability might not have completely been negated. And we didn't have enough patients to sufficiently segregate patients according to clinical subtype. For example, the presence of specific inflammatory drivers such as the shared epitope. So that's my talk. I'd like to thank my supervisors. These have really been excellent scientific mentors and they're excellent scientists and have given me a great deal of support and encouragement along the way. And I think with their guidance, I've grown a lot. And I think I'm very lucky to have had these supervisors. A number of people have been involved in this project and offered good support along the way. A committee, Hannah and Emma, Alex for bioinformatic analysis, Simon and Susanna and the patient donors, the PANG group and the Wix lab and the Hill lab. So it's been great to be a part of these groups and the inflammation division on the whole. Laura Dagley has been essential for the proteomics analysis and with assistance from Andrew Webb. Django and Phil have offered good advice in relation to size exclusion chromatography and Western blotting, as well as good life advice. Dennis, Tom and Kev in the instrument room. Eric for electron microscopy. Pete has assisted with figures. Laura and Dimitri for reagents. Betty, Madhu and Fig for Tim Tams. Mark Lee, Bioservice, the IT help desk, and Friday Soccer. My partner went to the UK more than a year ago, and Friday Soccer has really been the highlight of my week since then. So, and WEHI has really been just a fantastic place to come to and to study, and it's such a great environment here, and I think I'm always going to look back at my time here with great fondness. And finally, family and friends. Thank you.
Thanks, Andrew. Any questions?
So, Andrew, if you took the EVs from another disease state and you looked at the proteins, or for example, comparing to other studies, how different do you think that protein is in the EVs?
It's a good question. So we're kind of limited with synovial fluid because there's not too many diseases which need to have their joint fluid aspirated. The obvious one would be OA, and so that would be an interesting analysis to do and to look to see if there are less pro-inflammatory proteins and drivers of inflammation in patients with OA, which is typically a non-inflammatory arthritis.
Good talk, Ben. If you looked at taking those EVs and putting them onto cells and seeing if their responses are equivalent to the proteins that you've identified? So
that would be a good experiment to do and to find out, can we identify which proteins are responsible for, assuming they have a response, the cells have a response, which proteins are responsible for mediating that response. And no, we haven't investigated that in great depth. Sophia?
Kind of on the same line, if you look at the proteins actually in the synovial fluid, does it look like, you have the effects of what's in the EVs? So like, if you have IL-17 signaling, do you have IL-17 in the synovial fluid?
So that's a good question too. And we didn't see... We didn't really see a great number of cytokines in the EVs. We didn't look in this EV depleted synovial fluid for these cytokines. We did treat them with proteinase K, so you would imagine that if there were cytokines attached to membrane receptors on the EVs, they might be chopped off by the proteinase K treatment. one thing we were interested to see is, are there cytokines within the EVs, and we didn't detect a great deal of them. Does that answer your question? Probably doesn't too well.
You didn't look at the scenario fluid, so you don't know.
Kate?
Unless you want to come to. I mean, when you're looking at the EVs, your methods of looking at them, particularly with microparticles, which are butted for the membrane, a lot of known functions of those are related to what they actually have on the surface rather than what's necessarily in them. And your method is taking the microparticles and then cleaving whatever is on the surface. Do you think that this causes a problem in your analysis when you're looking at the vesicles, because you've essentially got rid of potentially half of what they would normally do in the situation? And is there any way that you think that you can counter that within your methodology, or is it just something that is a difficulty within?
Yes, so there are, one of the limitations is the proteinase K treatment, and we would have removed quite a number of the likely functional proteins on the surface of the EVs. However, it was a risk assessment. If we didn't do the proteinase K treatment, we would have a lot of this contaminating material. So I think in the interest of at least getting a snapshot of a large number of, an accurate snapshot of the large number of proteins within the EVs, we had to do it. But there would have been a loss, and an example of that is looking at the cell markers. It's quite, it's pretty unexpected for them to not be any EV proteins or to be very few EV proteins of macrophage origin or platelet origin. And these are cells which are present in the synovium or synovial fluid. Philip, babe?
So I was actually, I'm not quite sure to have understood the question is what would be the consistency or the reproducibility in the list of the proteins that you found, the constituents of your EVs that you found. If you were looking at 10 individual patients at about the same stage of disease. What I mean is that where your data accumulated, or where have you made this comparison between patients?
So I'm not quite sure what the question was.
Take the side of your fluid and analyze the content of the EVs in 10 different patients. How reproducible is that in the content?
So you're suggesting we compare, the synovial fluid compared to, on the whole, compared to synovial fluid with the EVs?
Just the EV content in 10 different patients.
How does it vary? Well, there was, you saw, we looked at the protein content with, there was a silver stain, if you remember, and it showed that there was very similar band patterns between individual patients with high inflammation and similar band patterns in between patients with low levels of inflammation. So,
my point is that in the take-home message for me, I'm not quite sure whether the EVs are anti-inflammatory or pro-inflammatory.
No, me neither. I think they're both. But based on the protein cargo, there was a surplus of pro-inflammatory proteins, and this suggests, it's not conclusive evidence, but it suggests they likely have a pro-inflammatory functional role. And this role, it probably depends on what their recipient cell types are as well. So the same EV might have vastly different effects depending on what cell they interact with.
So you've gathered a wide array of data here, and it seems, at least at face value, that a lot of the analytical approaches to deciphering that rely on methods such as ingenuity, and in the case of the microarrays, known targets and things like that. So all of those are really based on what we currently know about what targets are and about what pathways exist. And so I suppose my question is, if you just look at your data and you say, what's the top hit you've got here of a protein or an RNA that has not previously been associated with arthritis? What is that?
So I think that is a good approach, because it would be a good way to use this data to investigate novel things which are uncharacterised. We haven't really done that, and when it comes to writing this up, these will be things I guess I'll highlight. Ideally, we would would have lots of time. I could take these out and further go in and investigate exactly what these uncharacterized components of EVs are doing. But unfortunately, my time's coming to a close here, so I won't be able to do that. So I'll just have to pull them out and suggest that they might have important functional roles. Simon.
Thanks, Andrew. It's really good. In terms of trying to predict someone who's going to respond and not respond, I guess what you demonstrated is an average difference in the abundance of those RNAs. Did you get any sense, looking at, say, an individual RNA, the expression in an individual and how well it might predict whether they're going to respond or not, or combining a few of them together? I guess that's what we're looking for, is something to test and say yes or no, they're going to be responding.
No, it wasn't. It was all done as a group, so we didn't look at individual patients at an individual level. What's that?
In terms of identifying responders and non-responders as well, because this was like an RNA-seq-based approach, have other people also done like plasma or serum cytokine levels and looked at that and tried to use those to look at who would respond and who wouldn't?
Yes, Simon has. And if I remember, he didn't really find anything which really was indicative of response or non-response.
Yeah, I guess to follow that, so far there's nothing. So nothing from GWAS, nothing from all the genetics, nothing from measuring cytokines. So this is a big gap in what we can deliver. So what Andrew's doing, I started doing this 7 years ago. We didn't find anything then, and no one's found anything really compelling since.
I was speaking to a senior rheumatologist recently, and he was saying that the best indicator they have, anecdotally, is a patient's self-assessment score prior to commencing treatment. If they have a better self-assessment score, they're more likely to do better with treatment.
Andrew, there was a really great method for looking at the pooled EVs. Is there any way to... I don't know if you did this in your analysis, you looked at the differences of what cell types are producing the EVs. Is there a way you can check the differences between what cell types might be producing EVs more in the later stages of disease?
So that would require a longitudinal study with varying
I just
samples.
mean, even if you just compare your more severe disease with less severe disease, is there any indication that there's certain cell types that might be more represented there in the extracellular vesicles that are coming up?
Yes. So I guess comparing high and low, we did see that there were a greater representation of EVs from other immune cells, for example, T cells, but it was, I think it'd be nice to do this with a larger cohort, and the proteinase K treatment does present a bit of a limitation to it in that many of these lineage markers are membrane proteins and they might have been made invisible by the proteinase K treatment.
You've got this, it's more a theoretical question than anything, but you have increased inflammation in these patients and then you get these increased EVs that have pro-inflammatory factors. Do you think in fact that the EVs the body's coping mechanism to try and remove some of the pro-inflammatory signals, and that's why they end up there rather than the facilitators of the disease?
I think that is a good question, and so that there is good evidence that these EVs and the contents of these EVs are being taken up by recipient cells, but there is one school of thought that says these are essentially garbage disposals. The cells are releasing them to get rid of the stuff they don't need. So what do I think? I think they're there in high numbers, and I think I've seen good evidence that they are interacting with recipient cells. So I think they are functional, but there is still the possibility that in many cases maybe EVs from specific cell types might not be. We don't know. Leslie.
Now that you've given some more time to analyse your data sets, have you seen any correlation between your microRNA targets with your proteomics data?
That's a good question. So yes. So these are microRNAs which were decreased or in high disease activity or increased in low disease activity, and they correlated with the increased expression of specific proteins which were increased. So there is some suggestion that the microRNAs do affect the expression of specific proteins.
No further questions. It just remains really to thank Andrew. And if anyone does have any questions later, we'll be outside. I'm sure we'll take out and answer more questions. Thanks very much.